I wanted to go over how we're going to do this decoding RNA to protein and conducting a blast search activity. So we've talked about how to translate messenger RNA. Now I'm going to give you an actual practical reason why you do it. So we're going to be looking at the government's website, seeing where your tax dollars are going. Um, Probably a lot of people are looking at different proteins on the COVID-19 virus, so they can see stuff on that as well. There. Interesting website. Anyway, I emailed everybody a messenger RNA code so I would check your email, I would copy it, and then just paste it right down there, make things easy. First thing I would do is to change it into 3-letter codons. It's a little easier spacing it out there. Then you got to read the codon chart. So if you scroll down here, you'll find the codon chart. You can find the circular one online. If you don't know how to read this, watch the video that I attached to the weekly doc so you can go over it. Basically, you just start with the first letter here, second letter is up here, and the third letter will be whatever you end up with. So in our case, the first code was AUG, right there, AUG, which of course starts all proteins. And that's a way to kind of remember how to do that. You should know that AUG codes for methionine. There's only one code for met here, one methionine code, and that's right here. So, and you know it's AUG, and so it's A, first codon there, the U, the second codon, and the G is down here. So you kind of look at that to remind you how to do this. So it's AUG. The first one is methionine. GUA. So G and U. So we're in this box here. Doesn't look like it matters what the A is because they're all the same. That happens a lot in reading these. It's valine, so VAL. And then you would go on to the next one. I am just going to not bore you with that because I already wrote them up here. Let me just throw that up here. All right, so there they all are. Space them out right there. Okay. Now we're going to put these codes in NCBI. So let me just click onto that site. So go to that hot link. Now we're doing a translated RNA. So I have a protein code. But now I want a protein name. What is the name of that protein? So to do that, you see we got all these buttons here. And actually we could have taken that RNA code and used BLASTX, but we already did that. So we're going to go to protein BLAST, go here, and we are going to do the FASTA sequence. So the FASTA sequence is done not with these 3 letters. That's too many letters. It is set on the 1-letter code. So basically there's 20 amino acids, 26 letters in the English alphabet. So they have given a 1-letter code to everything. Often it is the first letter, like methionine is M, valine is V, but it doesn't always work that way. So tyrosine is Y, because there's a couple of T's. There's a lot of A, so alanine is A, asparagine is N, arginine is R. So you're going to have to check it out and look at it. Already have that coded right here. I'll just move that up here. All right, so that's the 1-letter code. And just to kind of see, so leucine was L, proline was P, but aspartic acid was D, glutamic acid's E, lysine is K, threonine is T. All right, so that's 10 letters. And when we put it in the FASTA, we can't have these spaces in there. all those spaces. And so on. I could continue, but I already have it right here. 10 letters is not enough really to find a protein, so I'm going to give you 30 letters to do that. So go to that and go to Protein Blast and get to the FASTA. I'm going to paste. So basically it's 10 letters, space, 10 letters, space, 10 letters, and you're only going to have 30 letters. Some of you may only have 29. When you get those in to that box, you're going to scroll down here to Blast and click the Blast button. And then we just wait a second or two or a minute. And we're still waiting. Oh, here we go. So now things have changed. The screen's changed, and we scroll down here. And you will see some names here. Where I want to go. Yeah, there's all kinds of names when you got only 30 bases. But if you notice, they all have something to do with hemoglobin or beta globin. This was a beta globin gene. The first one says something about hemoglobin. Another one about hemoglobin and hemoglobin. I'm pretty sure it was this beta globin one. So if you look at the percentages, though, 100% here, 100% there for that beta globin. So you can see the percentage of what it probably is. So it's Homo sapiens beta globin is what it was. So in the parentheses, that's the species it comes from. This protein here is where it's found. So that's the actual answer. So once you go to get that answer, you would just write beta globin. Then you're done. Hit submit, and you send me your code and your protein name, and that's all you do for decoding the RNA to protein.